So, that's a minute ago, so I give a couple words. I see.
You need to share to this share. Zoom.
Oh, you need to share with the Zoom. Okay, wait a second. So you have you guys think it's.
Yeah, I think it's not here yet. I can show the people here, but I'll just talk. So I can share my presentation afterwards to the people here. I just put this together because I was only asked a few minutes ago. I'm gonna give it a few words, but I wanted to tell you guys a little bit about my institute because it's a little bit unique in Canada. We're a federally legislated polytechnic, and so we're very much focused on applied research at BCIT. And we define this as advancing the state of practice. We think of this as being different than universities, which are really about pushing back the frontiers of knowledge. We're very much focused on solution-driven and working with industry and government. So my research department was founded in 1999 specifically to address industry and government research needs around product quality, safety, and efficacy. At this time, botanicals were being moved into a new regulatory framework in Canada called natural health products. And so there was a whole dearth of information to support industry in moving from what was sort of an unregulated area. Sometimes it was food, sometimes it was drug, into this new framework. And our metrics of success are a little different at my institute. So we're very much focused on the impact on policy, consumer health, industry improvement, presence in the marketplace, having student involvement to be more aware of what's out there, having multidisciplinary teams. My institute does not care if I publish at all. Like it's very different from academia in that way. They really don't care. They want to know that I can partner with industry, if I can help industry get products to market. I spend as much time working with universities as I do with industry because I'm often in the middle between the two. Right? So you have discoveries happening at university, but to translate that into actual practical things in the marketplace is sometimes difficult for them. So we do a lot of work in the areas of crop diversification, improving agricultural practices, doing evidence-based product development, improving products, processes, and a lot of regulatory compliance, both for Canada and the US. I think a lot of people are very concerned about it and confused about the different regulatory frameworks. So we spend a lot of time helping people pathfind the regulations and find their way to market. We do a lot of industry academic partnership. We really feel like this is important because our metrics are about solving problems and having a presence in the marketplace, but also employment for our students out of our programs. What I've learned over the years in working with industry is that it's very unhelpful to use project descriptions that are very vague, which is something that universities are very good at. You really need to have very concrete deliverables. And you need to be aware of the risks associated with regulatory pathways. I've seen a lot of academic researchers develop some amazing products that never made it to market because there was nowhere for them. A good example in Canada is a functional food. Like it just didn't fit anywhere, right? And so you can have this great product that ended up in a marketplace in Europe, but it had no pathway in North America. In the US, it was considered a food. And so, for example, a plant-based phytosterol margarine that's meant to reduce cholesterol is sitting next to a normal margarine in the grocery store, but costing 3 times as much, and no one knows why, because they can't explain why it's important, right? We work very closely with multidisciplinary teams because our students come from all different programs: biotechnology, food technology. We have students that come out of engineering. Business, we use business students as well to do market studies. We also work with fish and wildlife and sustainable conservation groups. I learned a long time ago that it's not that helpful to send a biotechnology student out into the field. They really get upset when they can't get the phone signal. I have pictures of students holding their laptops on top of mountains trying to get a signal. But the fish and wildlife students who are used to doing conservation work are happy to go out into the field and do plant collection. So that works much better. This is just one example of one of our projects. We've had a few different development projects, everything from getting wasabi into tissue culture and then into greenhouses. This one was the development of a novel beverage from a local Prof. Another one was working with the Vancouver Community College and the Culinary Institute. So we had baking students working with us, making bread with gluten-free bread and stuff. So it was really neat collaborations because having, bakers, plus they also brought us lots of other baked cookies and stuff. It was really fun. But we do a lot of that, and I think it's really important that our students get this opportunity to work in these very multidisciplinary teams. And it allows them to be creative. As I said, we do a lot of work on regulations. We do it across many different risk-based regulatory frameworks. We specialize in combination products as well. So we do have a ISO certified medical device group as well, as the biotechnology that focuses on drug development. In our department alone, I think we probably have 189 product licenses in Canada. Just from, I like to have my students go through the process so that they understand how you get a botanical product licensed in Canada. We do metabolomics. It actually started back in about 2000 when we were looking at traditional Chinese medicines and recognizing that we didn't have good quality control tools to support clinical trials. So that's when we really first started looking at using a combination of metabolite and chemical profile with chemometric algorithms. We really moved away from clinical trial characterizations and more into authentication for regulatory compliance using chemometric tools, and to be able to better differentiate closely related species and such as that. We focus heavily on validation of assays. So there are maybe 14, 18 official methods of analysis for dietary supplements in the US. 9 of them are ours from our lab. So we've been pretty active. We have official methods for everything from echinacea and ginseng to turmeric and Mitraganous speciosa, a plant indigenous to here in Indonesia. We've had funding for this work from everywhere from local BC Investment in Agriculture to the Ministry of Agriculture to the Agriculture and Agriculture Canada, but also from Health Canada itself, its regulatory compliance, and also the US through NIH and dietary supplements. We've also provided training in method validation worldwide. So for the USP, we trained government and researchers in Brazil, in Chile, as well as training through the American Society of Pharmacognosy and the AOHC. And I really hope to get to come to this with students and people here in the future, because we've provided a training manual as well. And our focus is to go from the field to our finished products. So we have a number of different projects that involve farm trials, often directly with producers. I really like working directly with producers, especially now as we have such an incredible impact on climate change, and we have a lot of species becoming endangered, especially ones that are in collection, not in cultivation. So more recently, our work has really focused more on getting wildcrafted materials into cultivation for long-term sustainability, but also looking at agricultural waste streams. Clinical trial, although we are doing a clinical trial on Mitraganous speciosa right now, which materials came from Indonesia, so I think that's pretty cool. And lastly, we have the same thing that everybody has. So we have except we have a food process resource center that allows us to do pilot scale manufacturing, including I have my own canning line and bottling line, so I can actually can beverages and test them. But we have all the usual equipment, except a few extra things like we have TXRF, which is as common, supercritical fluid extractors. Our NMI are certified juice and wine screening for regulatory compliance, which is a little bit different because that's our focus. And we have a fresh lab, our new food lab, which is certified by the Fraser Health Authority, so we can actually make food for consumption in our fresh lab. We also have 4 3D food printers. And that is it. So this is where our focus and capacity is. We like to do benchtop formulation all the way to commercialization. And I feel in this way, we have a lot of overlap with some of the work we've done at Brent, albeit ours is at a tiny scale compared to the incredibly impressive facilities and manpower and people and researchers who have here. So I'm really excited to be here. I'm so glad I'm here, and I think there's a lot of hopeful collaborations we can do in the future. That's it.
Thank you. Yeah, we will discuss which one. If some men already have to go to the mosque because it's Friday, but the ladies can stay here. So we have 2 questions. Question? Question to Professor Ilya. Sorry, could you elaborate more what it means by samples can be collected with minimal environmental impact and without destroying source plants? And number two, what is the error value in the result of range method compared to laboratory test standards, both in conventional and algorithmic method? Do you want to answer that?
Sure. Well, yes. The conventional ways of bioprospecting and collection, the conventional way of bioprospecting usually involve uprooting the whole plant and then drying it. And so this has environmental impact because you remove the plant, because you need a lot of material to do your large laboratory extraction. When you only need 2 grams of tissue, you can take half of the leaf, and let's say you collect half of the young leaf, half of the old leaf, and those are your samples. So plant is perfectly happy if you clip half of its leaf and it has another 300 leaves. So that's why it's such a minimal environmental destruction. Your plant perfectly survives. Sort of like doing a blood test on human being, pretty much, right? You don't have to kill a plant. So that's answer to the first question, just because we miniaturize basically everything. And the second one was what? Yeah, well, error value is really, unfortunately in science, often is a function of a person rather than a method. So that's a very difficult question to answer. And I think one important thing we need to do more is really validate RAMS technology with a known compound. And particularly, we've done some work showing that they're very stable. That actually, biochemicals, because they're so rapidly preserved and dried, they're very stable. But we would like to do much more to show that actually the stability of extracts on glass fiber discs, for example, is higher, which we believe we see then if you store them in vials as a sort of tarry substances, which often happens, exposed to air and never totally dry. So there's a lot of work to be done, but we do think that actually what we see so far is very little error because it's such a rapid and very gentle way of preserving your compound.
Okay, thank you. This one is a question from Malam. I invited many, not just research center of genetic engineering, but also center for botany, sitting at the back there too, and then center for natural product chemistry, and also from other university joining this Zoom meeting here. So thank you for the answer. Vyacheslav, I hope it's clear enough for you. I was we can discuss. Next one, and also a question to Professor Dushenkov. What is your view on the opinion of researchers regarding the publication of medicinal plants recommended should be organic farming? Or some researchers argue that a more sustainable way is to way to the production of secondary metabolites from medicinal plant, preferably with biotechnology and precision agriculture, such as agriculture, mass production using bioreactors, and greenhouses.
Whenever you're talking about the bringing medicinal plants to the market, one of the things you have to consider is the whether there is a sufficient source of the plant material. In many cases, if it's a new plant, it started with collecting in the wild, but it's not sustainable. You always have to think and figure out whether you can bring the plant into the culture. And for the ferula plants, for example, it was very successful. There are at least 2 different species of ferula, which is grown right now as a cultured plant. And for those 2 species, what they use is the gum which produced by the roots. So it's like harvesting every year. How that looks like for Pirula, for example, they're grown in the specific places, and then you sort of dig a little bit of the roots, expose them, and damage them just cutting a little bit of the roots. Then you create the gum which is coming out of the roots, and that's what's used to produce the spices or medicinal material. Then the plants get to rest for a couple of years, and then you go to the next one. So in my opinion, the sustainable use of medicinal plants is getting, especially with a higher volume, only through the cultivation. However, at the beginning, when you're just starting to do the research and starting even the initial production, it could be a sustainable harvested from the wild. And depending on the situation, you have to evaluate the total volumes of the medicinal plants that you can harvest, and whether it can be done sustainable with the benefits to the local community. In some cases, indigenous community can provide a sufficient amount of this plant material for initial stages of marketing without damaging the actual population of the mound.
Thank you, Mr. Dushenkov. This one more moving six. Okay, he just said that. Thank you so much for these words from both of you. So, Prof. Awan, yeah, Prof. Awan would like to ask you regarding.
Very interesting presentation. I would like to know if you have a kind of measure or focus of kind of plant crops and also the substance that you're running in your facility. I mean, maybe you have the major program or something like a vaccine in your facility.
So our projects come entirely from government or industry, right? So we don't. I've almost in no case have I ever just selected which plant I want to work on. I do have as a Canada Research Chair holder, I do have a lot of discretionary funding that does allow me to do a little more basic research and a little more investigator-driven research. But for the most part, it comes from, we look at the government priorities. We look at priorities of the agriculture in the region of British Columbia, but as well as federally. Or we look at what is a priority to our industry partners. So for that reason, kind of every project is unique. Okay, so if we're doing sectoral good, it's because we have an entire group of producers that are having a problem, right? Like starting way back in 1989, ginseng, American ginseng, used to be one of the largest crops in British Columbia. It was the highest grossing crop, but they were having, issues with pests, and they were having challenges with contract labs getting consistent data. So that's when we came in to do research on ginseng, right? That kind of led to working with Environment Canada on situs enforcement, because we have a lot of Panax Cornicifolius being mixed up with Panax ginseng, whether it's regulated, when it's not. So a lot of that kind of work is what we end up doing. It really just depends. Yeah, we have to be nimble.
Yeah, you don't have a roadmap, something that you play as your specialist or something?
Well, in Canada, our specialty is natural health products. So we're one of the few research groups in Canada that does specifically natural health product and dietary supplement type research. We don't really have a tradition of pharmacognosy in Canada. We don't have schools, we don't have people like larger groups, like at Rutgers, like they have in some of the schools. And so there's little bits of researchers out of food, out of pharmaceutical sciences, but we're the only research team that's entirely devoted to helping get in traditional medicines to market.
Thank you.
Okay, thank you. So I think, because of the time, otherwise it would be very interesting. We can go on and on until the end of the day, because, we're very fortunate with these three, having this scientists and prominent scientists also in the world. So thank you very much again. I hope the collaboration, of course, with Prof and CUNY maintain and intensify while with Columbia Institute of Technology, just starting from now on, but I hope we can collaborate also. Yes, thank you very much again. All the audience until now, still how many people in here except some actually it's 11:45, so we can still run to the mosque. So thank you again. I hope we can meet again because I cannot really stop the discussion because of only the time that can be discussed. Okay, thank you, Burari. It's actually back to you. Thank you very much for the hospitality and also for assisting and making this seminar possible today. Thank you, because one or two sentences, but okay, back to MC. Yeah.
Okay. Thank you, Prof. Annie, for leading a very interactive discussion. And thank you for the speakers for a very interesting presentation. Ladies and gentlemen, this is the end of our agenda for today. And on behalf of the committee, I would like to say thank you very much to our speakers, moderator, and all participants. See you in the next event. Wassalamualaikum warahmatullahi wabarakatuh. Thank you.
Thank you.